Good afternoon, everyone. Thank you all for coming. My name is Anish. I'm a PhD student at the University of Washington, Department of Pharmaceutics, and my talk is titled The Assessment of Risk Factors in Chronic Kidney Disease Using Proximal-Tubule MPS. And today, the specific risk factor I'm going to talk to you about is a food contaminant and mycotoxin called ochratoxin A, shown here. So first of all, chronic kidney disease is a slow and progressive condition which involves structural and functional decline in the kidney, which results in reduced filtration of the blood. And the key pathophysiology of this disease is high proteinuria and a high degree of glomerular sclerosis. And about 35 million people in the United States suffer from this disease, and about another 35 million people in Europe as well. So some of the main risk factors in this disease include diabetes, hypertension, and obesity, but importantly, acute kidney injury, which can result from things like exposure to toxins, abusing NSAIDs or immunosuppressants or infections can lead to this disease. And importantly, a single instance of acute kidney injury can increase your risk of developing chronic kidney disease by tenfold. So it's important to note that actually half of the world's CKD cases and deaths occur in the developing world. And in the last 30 years, health professionals in these regions have been noticing that a large proportion of the patients otherwise seem healthy, but they don't have the traditional risk factors that I mentioned in the previous slide, but they still present in the clinic with end-stage renal failure. And so this condition has been termed chronic kidney disease of unknown etiology, or CKDU. And as the name suggests, it's not really known what causes this condition, and it's highly debated in the literature. And one important distinction that I want to make between CKDU and CKD is that in this condition, there's a high degree of proximal tubule damage rather than glomerular damage. And so as this disease normally affects younger men who live in rural agricultural communities, it's thought that a multifactorial set of environmental agents might be at play here. So these are things like pesticides, herbicides, heavy metal contamination in the groundwater, things like lead, arsenic, cadmium, heat stress, and mycotoxins. And also I'd like to point out that in some of these regions, such as in India, Sri Lanka, Mexico, this condition accounts for upwards of 70% of all CKD cases. And now one such mycotoxin, as I mentioned at the beginning, ochratoxin A, has been suggested to be linked with several of these CKDU endemic nephropathies, such as in Sri Lanka. So this mycotoxin is produced by the Aspergillus and Penicillium genera of fungi, which grow well in tropical environments, but is found throughout the world. And now ochratoxin has a high carcinogenic potential. It's a known nephrotoxin and a known hepatotoxin. And the most commonly contaminated types of food are usually grain products such as wheat, rice, wine, coffee, beer. And concerningly, only a few governmental agencies around the world actually regulate ochratoxin contamination in food. And you can see here the threshold set of about 10 micrograms per kilogram based on food type. And now to give you an idea of how contaminated some food products can be, I have here a table of import rejections from 2015. And you can see the high degree of contamination, upwards of 2,000-fold higher than the levels set by certain governments. And it's been estimated that the average human serum concentration worldwide for ochratoxin is about 2 nanomolar. And in fact, in a CKDU patient found in 2003, the highest recorded serum concentration was 5.6 micromolar. And concerningly, for kidney health, ochratoxin has been shown to accumulate in this organ. In rat models, it was found that ochratoxin levels in the kidney were 12-fold that of plasma. And additionally, in vitro assays have shown that OTA has a high affinity for the proximal tubular uptake transporters OATS1 and 3, and a low affinity for the efflux transporters such as BCRP and MRP2. And even worse, ochratoxin has a high affinity for the apical reuptake transporters OAT4 and PEP2. So essentially what this means is that there is a high efficiency uptake or transport of ochratoxin from the blood or from the urinary filtrate into the tubule and not much being secreted. So you can see how this might lead to accumulation, toxicity, and disease. And to kind of further highlight the prevalence of this toxin, we actually analyzed reservoir samples from endemic and non-endemic regions of Sri Lanka looking for OTA in the groundwater. So we found levels of ochratoxin of about from 0 to about 20 picograms per milliliter. And this analysis is currently ongoing, but what I really want to highlight here is that we also found detectable levels of ochratoxin back home in Seattle, where our lab is, in the bay behind our lab, and concerningly in our tap water. So this kind of goes to show the importance of studying this toxin and its relevance for human disease. So our overarching hypothesis here is that OTA targets the proximal tubule. Accumulation and its nephrotoxic effects may contribute to CKDU disease progression. And now the significance of ochratoxin exposure in human health remains unclear, and this is in terms of its biochemical mechanisms of toxicity, its metabolic fate in the human body, and subsequent toxicokinetics, and of course its overall relevance to CKDU. And these 3 points here are essentially the 3 main aims of my thesis. And today I'm going to be talking to you about this first one, where I'll characterizing its biochemical pathways in proximal tubule and my use of MPS to investigate this. So the system that our lab uses is the Nordis Parvivo triple channel chip, shown over here. And we isolate primary human proximal tubule epithelial cells from whole kidneys and biopsy samples, which we then seed and grow out, and then seed into the collagen matrix of the chip device, in which the cells form a tubular monolayer. And then when these are hooked up to, pneumatic pumps, we can perfuse cell media unidirectionally through the tubule, which allows for compound or test compound administration and analysis of the effluent that flows out. And of course, our lab has demonstrated that these systems recapitulate in vivo proximal tubule function not seen in 2D, such as proper localization, polarization, and function of transporters such as the OATs and BCRP, MRP. So some background on what's known with OTA and some initial experiments that our lab has done. We've found that ochratoxin downregulates the mRNA of several downstream targets of the transcription factor NRF2, shown down here. Now NRF2 is the master regulator of the antioxidant response, and under oxidative stress, it translocates to the nucleus and activates the transcription of several enzymes involved in things like the glutathione or the thioredoxin cycles, very important for regulating reactive oxygen species. And so it's not really known how ochratoxin does this. It's hypothesized that it may inhibit the release of NRF2 from its cytosolic binder and ubiquitin ligase, KEAP1. So to investigate this, I simply treated our PTEX in 2D with ochratoxin and 2 NRF2 activators, sulforaphane and TVHQ, and performed mRNA sequencing. And so essentially here we can see the downregulation of several NRF2 targets. I have here glutathione reductase, GST-pi, and in this case we can see significant induction of this enzyme with the NRF2 activation, but essentially this is completely reverted with co-administration of OTA. And essentially, this data suggests that there's likely not a direct interaction with ochratoxin and the NRF2 KEAP1 complex. But what's really interesting is that in this data, we found significant upregulation of the hypoxic response via HIF1 alpha and several of its downstream targets in our cells due to OTA. And so some of its downstream targets, such as PDK1, lactic dehydrogenase, facilitate a metabolic shift in the cells from oxidative phosphorylation to glycolysis. Now this is really important because the renal cortex, the proximal tubules, utilize about 20% of the body's oxygen to produce ATP, which is essential for maintaining the ion balance that facilitates fluid balance and active secretion. And so the PTEX are very susceptible to hypoxic stress. This was corroborated by immunocytochemistry staining for HIF1 alpha in our MPS. So here we use cobalt chloride, which is a chemical inducer of HIF1 alpha, to simulate low oxygen conditions and hypoxic stress. And here, you can see that a sort of synergistic effect between ochratoxin treatment and our low oxygen conditions on HIF-1 alpha nuclear stabilization, and also you can see cytotoxicity, significant cytotoxicity with the nuclear stain, and over here you can see disruption in the tubule. And so again, highlighting the susceptibility of the proximal tubule to low oxygen. So kind of going back to that mRNA data and building off of this, we also noticed several of the key regulators in mitochondrial fusion, fission, and mitophagy were dysregulated due to ochratoxin treatment. And now this is again very significant, again because, as you know, oxidative phosphorylation occurs in the mitochondria, and then proper regulation of these processes remove damaged mitochondria, help keep the reactive oxygen species low that are produced as a result. And again, ATP production is very necessary for proximal tubule health and function. So moving past transcriptional analysis, we can see this mitochondrial disruption phenotype in our cells in 2D culture with confocal imaging. And here we labeled the mitochondrial membrane with GFP. And so in healthy control cells, you can see that the mitochondria sort of form a elongated, interconnected network. And with our treatments under low oxygen conditions, under hypoxic stress, you can see a breakdown of this healthy network. And even at physiologically relevant concentrations of OTA, we can see a similar breakdown in the mitochondrial network. And so this 2D imaging data was analyzed in ImageJ with a script developed by the Valente group. And it essentially shows a breakdown in mitochondrial network mass, shown on the left, and a decrease in mitochondrial complexity. This here is looking at the branches per network, or the complexity of the network structure. And similarly, an increase in the amount of individual structures compared to more complex networks. So essentially, this is all showing an increase in fragmentation of the mitochondrial network due to OTA treatment. And I'd really like to highlight that mitochondrial fragmentation has been associated with other forms of kidney disease, such as in diabetic nephropathy, and also other chronic diseases such as liver disease and Parkinson's. So can we see these can we see this in our chip systems? So obtaining high-resolution subcellular images in microfluidic devices can be a challenge, as the specimens are usually larger, and the complex structure of the tubules is not optimal for confocal microscopy. However, with spinning disk confocal, you can see this technique offers an improved imaging depth and high-speed acquisition of large structures. And so with that, we were actually able to obtain high-resolution subcellular images of our tubules, and we see the same phenomenon of the breakdown of the mitochondrial network. Now, quantitative analysis of 3D images from these devices are also another challenge. So the script I showed you earlier from the 2D data does not work with 3D images. However, in the imaging software Ameris, which some of you may be familiar with, the most recent update incorporates artificial intelligence and machine learning to one of its modules called the filament tracer. Now, this was originally developed to analyze the structure of neurons, but with this new improvement, it can actually be trained to recognize subcellular structures such as microtubules or mitochondrial network. So I'm basically showing here its capability to recognize and reconstruct the mitochondrial network in a 3D image. Now, this is very preliminary data. This is the only one I have because I did this about 2 days before my flight over here. But yeah, so this tool, along with spinning disk confocal microscopy, can be a powerful tool for imaging subcellular structures in complex systems such as MPS. So to summarize these findings, I first showed the downregulation of NRF2 target genes and its effect on the, OTA's effect on the antioxidant response is likely not due to an interaction with its cytosolic binder KEAP1. And I also showed that ochratoxin induces the hypoxic response both in transcriptional data and in our MPS. And similarly, ochratoxin induces mitochondrial dysfunction, which was shown with mRNA data in our 2D confocal imaging and in MPS. So again, I'd like to highlight that dysregulation of mitochondria is associated with a number of chronic diseases, including acute kidney injury, or the progression of acute kidney injury to chronic kidney disease. And so some future directions that I'd like to briefly highlight are, so we're seeing mitochondrial damage in our cells. This may lead to a production of pro-inflammatory cytokines via the cGAS sting pathway, which we plan to investigate using our MPS and the effluents that come out of the chips. And notably, a future experiment we have planned is to characterize the renal clearance of OTA using a dual-channel vascular proximal-tubule chip system and use the data from the transport study to develop a physiologically based pharmacokinetic model of OTA to understand its disposition and accumulation and clearance in humans. So yeah, with that, I would like to thank my lab, everyone involved, my advisor, Dr. Edward Kelly, as well as our collaborators at the University of Pittsburgh and Stanford and in Sri Lanka, and my funding, and of course the MPS World Summit Organizing Committee for inviting me to come out here to Berlin to talk to you about my work.
Thank you for that great talk and you shedding new light on ochratoxin, which is a very old toxin known for ages. Yeah. Are there any questions?
Very nice talk. Thank you for the experiment. I have actually two questions. The first question is, have you ever measured the in situ production of glutathione and also the gene expression of the glutathione producing genes? And the second one, is there any data, clinical data or epidemiological data showing the urine concentration of ochratoxin for the farmers or something?
So to answer your first question, yes, with ochratoxin, several of the genes that are involved in glutathione production, such as like GCLC, were heavily downregulated. Also, the transporter, I believe it's SLC7A11, which takes cysteine into the cell to make glutathione, was also very much downregulated. I didn't quite hear your second question. Could you please repeat?
Is there any information for the urine concentration of ochratoxin measured in epidemiological data, like in the farmer or some people who are exposed to them?
There's not much. So the studies that I showed where, do I have it? Okay, I don't have it on this presentation, but yeah, so serum concentrations of ochratoxin have been shown to be around like 10 nanomolar in like CKDU patients. It's very variable, which is why it's like ochratoxin to CKDU has just been proposed. It's not like there's no like clear link. In some cases, you'll see higher levels of ochratoxin in control or healthy patients versus in other regions of the world. In the CKDU population, you can see much lower. But yes, as far as the range goes, it's around 10 nanomolar to 50 to 100 in some of the disease studies.
Very nice talk. I have also two questions. So one is, have you ever seen these lysosomal dysfunction, for instance, by the LAMP1 expression? And the second question is, do you consider these lumen side uptake as a primary?
So lysosomal, we haven't seen lysosomal dysfunction, but it is something we're thinking about because that could very well be an active player in its toxicity. And in similar diseases such as like cisplatin-induced nephrotoxicity, that's also been shown. So that is something we're going to look at.
Thank you very much for the nice talk. I would also have two questions. The first is, you said you have been isolating the proximal tubular cells from whole kidney.
So I would be interested in how you did this, which isolation technique you used, and how you could prove you have 100% pure proximal-tubular cells there. And the second one, I don't know, perhaps I have missed the point. Would you think that your model would be also feasible to have several dosage of the toxin into the model, or is it just for a single application? Thank you.
So for your first part, to isolate the proximal tubule cells from whole kidneys, we essentially dice the renal cortex, and then through centrifugation with collagenase and dispase, we isolate the proximal tubule cells, and we have done flow cytometry analysis to see if they're actually proximal tubules, and we see about 80% proximal tubules and about 20% distal or convoluted tubules. So for different concentrations, we usually just have like, one tubule in a chip would be like one concentration, and then, differ it that way. Is that what you're asking?
Right. I'm curious about the mitochondrial dynamics. So you showed there's a lot more fragmentation. Is it just a shift from, like the longer mitochondria to more the smaller bits, or is there a reduction in total mitochondria? Like, did you measure total intensities?
Yeah, so both of those things. So you can see here, hopefully you can see it. It's like elongated tubule fibers in a healthy mitochondria where fission and fusion are balanced. And then when you have a toxin or condition that disrupts that balance, most often in diseases you see fragmentation or a higher degree of these individual dot-like structures.
Did you look at autophagy markers too? Like, is there any connection between autophagy and mitochondrial fission?
So that's theorized. We haven't looked at it yet, but I plan on doing a flow cytometry analysis for looking at that.
Thank you.
Thanks, Anish.
Thank you.